Hey everybody, welcome to chapter 15. I realize I'm starting on the wrong the wrong slide. Here we go. Chapter 15 is all about adaptive immunity. It's the other branch of our immune system that is the corollary to the innate immune system. So again, this is a fairly heavy chapter in terms of learning objectives, and there is a lot of content in this unit. I'm sorry, in this lecture. So let's go ahead and dive right in. Remember that the innate immune response, when you think of the innate immune response, you should kind of think about, oh goodness, I don't know, what are the characteristics of the innate immune response? That'd be a nice, like, kind of get class started question if we were meeting in person. So let's think about so innate immunity is common, kind of conserved across multicellular organisms. So really our ancient ancestors had this, and organisms that were barely related to still have the innate immunity. It's fast acting. It's usually the first kind of part of our immune system to respond to an infection. And it's also general. Remember, it recognizes patterns. It's really a non-specific kind of response to infection. Remember that once, for example, neutrophils get activated, they just start killing everything. They just kind of go crazy, including themselves, right? They last for... I think they only last for like a couple days, and then they basically work so hard that they die, and they cause a lot of damage in our body. And the idea is that they're causing more damage, of course, to the pathogen. But in contrast to the innate immune response, the adaptive immune response is really, oh my goodness, got another call. Oh my goodness. Let me hold on a second. I've been getting text all day because of grades. Hold on. Okay, sorry about that. I'm back. I have, ever since I sent out, so I have this class and I have a 107 class that has like, I don't know, at least 50 students in it. And they, ever since with the deadline for dropping approaching, I'm just getting call after call and text after text and all these emails asking for their grades. And I can't get anything done. I'm so behind. So anyway, enough about me whining. So let's talk about the adaptive immunity and what are the characteristics of adaptive immunity. Remember that, so the three things, and this is definitely a big part of the exam, the three things that define the adaptive immune response are specificity, memory, and tolerance. Adaptive immune response is highly, highly specific because it selects for cells that will only recognize very, very specific patterns on specific pathogens. And it turns out that we can actually, it's so specific that we can actually use this as a tool. And we'll talk a little bit more about this when we talk about antibodies. And in the last unit, we'll talk about applications of immunity. So highly specific. If you get, so for example, a perfect example of this is that we've all been infected with the coronavirus at some point in our lives, probably many of them. But none of us have immunity to COVID-19 because we haven't seen it yet. So as very closely related as all of those viruses are, because they're ever so slightly different, we probably, I say probably because we don't know for sure, but we probably don't have immunity to this COVID-19, COVID-19, I don't know, for some reason that didn't sound right, COVID-19 virus that is, of course, making its way around the world. The second thing that defines the adaptive immune response is memory. The adaptive immune response remembers pathogens and is better equipped to fight them off the second time around. This is basically what makes vaccination possible. And again, we'll talk about this in Unit 5. This is in contrast again to the innate immune response. The innate immune response just is. It gets activated and it just goes, and it doesn't really, there's no difference between the way our innate immune system functioned when we were 5 years old versus 10 years old versus 50. It's just a part of our, it's encoded in our DNA and they don't change. Whereas our adaptive immune response changes all the time. And then finally, tolerance. So tolerance basically in a biological or immunological context basically means that it's able to tolerate self. In other words, it doesn't recognize or harm, or at least it's not supposed to recognize or harm self cells. And if it does, we have an autoimmunity problem. The adaptive immune response can be further divided into two major parts. That's the humoral branch of immunity, or humoral immunity. When you think of humoral immunity, you should think of antibodies. So the humoral branch of the adaptive immune response is really mediated by antibodies, particularly antibodies that have been secreted by B cells. So B cells are really the main effector cell of humoral immunity. Whereas in cellular or cell mediated immunity, it is really mediated via immune cells of our system. Specifically, in my opinion, it's mostly cytotoxic T cells. There are certainly other cells that play into this branch, but when you think of humoral mediated immunity, you really should think about B cells. And when you think about cellular immunity, you should think about cytotoxic T cells. And that's not to say that that's it, but that's the main player. That's kind of the main effector cell for each of these branches. And they also have a difference in what they target. So the humoral immune response typically targets extracellular antigen. So if we have bacteria in our bloodstream, bacteremia or septicemia or something like this, it would be handled mostly by our humoral immune response. Whereas if we have cell damage or cancer or harm that is happening within our cell, or a viral infection, for example, if the virus is inside of our cells already, it's not outside of our cells, then that response is mostly mediated by these cytotoxic T cells and our cell-mediated immunity. So I thought I'd leave some of these questions in, even though, well, I guess I can discuss them. These would be the questions that we would discuss in class kind of to get lecture started. Remember that dendritic cells and macrophages kind of serve as a bridge between the innate and adaptive immune systems. They kind of have a cross function. One, they're phagocytes, and they're usually some of the first cells that will migrate to the site of infection. But then they have a further role in the adaptive immune response because they're going to carry those pathogens to our lymphoid organs and present those pathogens to our B cells and our T cells to educate them about what's actually happening and get them ready to go to war. If you recall, we also had a big flow chart of all the different cells of our immune system, and I want to point out that there's only a very specific subset of our cells that are associated with adaptive immunity, and they're called lymphoid cells. And there were 3 cells that make up this category. There were B cells and T cells and B cells. And the one that doesn't belong, and that's natural killer cells, NK cells. I'm gonna look up the diagram really quick. So we have, so our lymphocytes are B cells and T cells, which are absolutely part of the adaptive immune response. But then we also have natural killer cells, which are not adaptive immunity. It's really important to point out that NK cells or natural killer cells are really part of the innate immune response. We kind of lump them into this chapter because they're a lymphoid cell, but I really want to point out that they are they're actually part of the innate immune system because they're non-specific, right? It's that specificity and that memory that makes it adaptive, and natural killer cells don't have that. So we'll talk more about that later. Okay, so let's talk about how we can further kind of differentiate between adaptive immunity and, humoral immunity and cell-mediated immunity. So here we have in the pink, here's our humoral branch, and here's our cell-mediated branch. Notice that I said the main effector cell for humoral immunity is B cells. For T cell, or for cell-mediated immunity, it is T cells, specifically cytotoxic T cells. I very much feel that helper T cells are both. They help both humoral and cell-mediated, but because they're T cell, people tend to lump them in with cell-mediated response. So this is a nice, I like this picture because it gives a nice overview of what is actually happening. And if you can come back to this picture at the end of this lecture and kind of understand what's happening, you should feel good about what you're doing with this chapter. So just to kind of give a brief overview of what can happen here. remember that we have this bridge cell called the dendritic cell that is, even though it belongs to the innate immune response, it really has it. It's interesting for me to say that because I've always thought of dendritic cells as part of the adaptive immune response because they play such a critical role, but they really are part of both. So they have a function in innate immunity that we've already talked about, but they really have a major role in adaptive immunity because they're the cells that are going to wake up the B cells and the T cells. And they do this in the case of T cells, they do this directly by simulating them via pathways that we're going to talk about. And what that does is it causes them to mature from a naive T cell into what their actual function is. And T cells have two major categories. We have helper T cells, so that H stands for helper. And we also have T cells that are considered cytotoxic cells. So the C stands for cytotoxic. So what happens here is these cytotoxic T cells can go to the site of infection or the site of injury and kill cells directly. They deliver messages and stimulatory proteins to that cell that cause it to kill itself, undergo apoptosis, or it more directly will dump some enzymes and some kind of really toxic, they're called granzymes and perforins, that will kill that cell directly. Now, on the other hand, we have these helper T cells that really, in my opinion, help kind of both branches. So the helper T cells are going to deliver signals, so they help, right? They're not going to do any killing directly, but what they really do is they deliver amplifying messages in the form of cytokines to other cells, namely macrophages and B cells. So when they deliver the signal to a B cell, it's going to cause that, it's one of the signals that's going to cause that naive B cell to mature into a plasma cell. And as we will learn here very shortly, plasma cells have one job, and that is to produce antibodies. Yet another call, really quick. I should just turn off my ringer. Hold on, let me turn it off. And remember that in the humoral immune response, those antibodies are going to basically take care of any extracellular antigen, like in this case, a bacteria. They're going to surround it, neutralize it, and eventually cause it to be phagocytized by like a macrophage or something else. So we have our three main kind of effector cells of adaptive immunity. We have B cells, we have helper T cells, and we have cytotoxic T cells. So notice that they all have kind of a naive stage where they're basically resting. All of us, hopefully, if we have no active infections right now, we have no kind of angry differentiated B cells or T cells right now. If we do, we might see some plasma cells in our system, or some active helper T cells or cytotoxic T cells. All right, so let's get into some of the structures that are involved in our adaptive immune system, specifically the lymph system. The adaptive immune system, or the adaptive immune response, involves a lymphatic system. We have two major categories. We have the primary and secondary lymphoid organs. Our primary lymphoid organs are red bone marrow and our thymus. Our bone marrow is found in our hollow bones, mostly our femur. Our femur is a major source not only of those hematopoietic stem cells, but also the differentiated lymphoid cells. And our thymus is right above our heart. It's a tiny little guy. It's actually quite small. And so what is it? It's like the size, I want to say it's like the size of a walnut in children. It's never that big. Let me Google this. I forget how big a thymus is. How big is a thymus? Let's see here. So at birth, it's 4 to 6 centimeters long and 2 to 5 centimeters wide. So that's about what? 1 by 3 ish. So yeah, about the size of a walnut, like maybe a golf ball size, approximately. So it's not very big, and that's at birth. And what actually happens is it continues to shrink. In fact, by the time you're an adult, it's really small. And I want to say in senior citizens, you can't even find it because it plays a major role when we're young in the education of our T cells. And as it kind of gets disused, actually what ends up happening is I want to say that our sex hormones, basically as we reach puberty, that avalanche of sex hormones that get secreted into our body give it signals to start to atrophy and go away. And so by the time you're old, you can't even find it anymore. I think you have to use you can find tissue remnants of it or something, but it basically just goes away. Our secondary lymphoid organs are our spleen, our lymph nodes, and a really interesting association of lymphoid tissues called the MALT, which is mucosa-associated lymphoid tissues. It kind of surrounds our stomach, and they include the Peyer's patches, and it has some really interesting functions with gut-associated immunotolerance. So there's kind of this idea in immunity, like if you eat a lot of antigens, then that somehow teaches your body how to tolerate those. Basically you'd become less allergic. So if you eat, there's an idea that if you eat local honey, for example, eating that local honey, it contains pollen from all of the different flowers and whatnot that you will most likely find in your area. And eating that pollen through the honey somehow educates your immune system that this is safe and it's not going to cause a problem for you. And that is mediated through that mucosa-associated lymphoid tissue. It's a really cool avenue of research for basically handling allergies. But as with most things in immunology, it's super complicated and hard to study because it involves so many working parts. Okay. All right, so let's characterize some of the major players here. Let's first talk about B cells. Remember that when you think of B cells, you should think of humoral immune response. B cells are born in our bone marrow. Remember that they are derived from our hematopoietic stem cells. And they undergo a process, how should I say this? Okay, so we'll talk about the, let me come back to what clonal deletion is. Let me talk a little bit about B cells first. So the B cells ultimately are the cells that secrete antibodies. They're the only cell in our body that actually does this. So they perform a very important function. And again, they're most effective against extracellular antigens. So bacteria when they're outside of our cells, toxins that are secreted by those bacteria, and viruses that are in the external environment. In other words, they're not inside a cell. So they have markers on their membrane. We're going to call it. So there's all kinds of markers on the outside of the outside of the cell. And let me pause here for a moment and talk about how we talk about these markers, because it's a very confusing topic in immunology. We call them CD markers. So CD stands for cell determinant. And so some of these cellular determinant markers are common enough that we actually have a name for them. So like a B cell markers, or the B cell receptor, the BCR, and we call them by our names. Whereas other cell determinant markers, we simply say CD, CD4, CD8, CD95, and so on. So there's numbers associated with them. So if you ever see that CD kind of abbreviation, it's simply talking about a marker, or usually a protein or a glycoprotein that's on the outside of a cell that lets other cells know what that cell is. So there's characteristic markers on the outside of every particular kind of cell. So these B cells, for example, have many markers on the outside that signify that they're a B cell. And then they also, very importantly, have this B cell receptor. And the B cell receptor is going to determine the type of antibody that it can ultimately produce, because it's that protein is the antibody, but it's just embedded in the cell membrane of the cell. So, yeah, hang on to clonal deletion. I think there's a better place to talk about what clonal deletion is later in the unit. So I'm going to make a note to myself too, so I don't forget to talk about it. Let me just grab a piece of paper here. Okay. B cells will eventually differentiate into plasma cells. Plasma cells are professional antibody production B cells. So when a B cell gets turned on, in other words, when it gets activated by cytokine signals and contact with an antigen, this is going to cause that B cell to differentiate into a plasma cell. And the job of a plasma cell is to secrete antibodies like crazy. Like they just go, and they're kind of like neutrophils at this point, except that they're not secreting little hand grenades everywhere. They're secreting antibodies, and they secrete just prolific amounts of antibodies, and then they die. So they really can't last that long, because they basically, once they get turned on, they go until they get exhausted, and then they just kind of give up, and they die. And we actually want that. I mean, if you think about it, we want to clear most infections that we have, we clear within a few days. And so we don't want these plasma cells just hanging out forever and constantly secreting these huge amount of antibodies, because that's going to have an amplifying effect throughout our body. So we really want our plasma cells to be short-lived, so that they're not, like we just don't have a constant immune response. However, the antibodies, depending on the kind of antibody that it secretes, those antibodies can stick around for quite a long time. And this is why it's so important, this on a side note, this is why it's so very important that we get that antibody testing for coronavirus up and ready as soon as possible. Because what's going to end up happening is a lot of people are going to test negative for it that are actually positive, because these antibodies only stick around for about a month at most. So if any of us had coronavirus back in January or February, when people are hypothesizing that we kind of had this initial wave of coronaviruses, chances are that those antibodies are already gone out of our system. And if we got tested, it would be very difficult for that test to detect positive antibodies in our system. And so we would have, what would that be? A false negative, right? A false negative. All right. Now, a small subset of these B cells, instead of turning into a plasma cell, they turn into what we're going to call memory B cells. This is that memory function of our adaptive immune response. And essentially what these do is they don't get activated. They don't turn into these antibody production factories. What they do is they remember. They hang out and they live for a very long time. They usually hang out in our secondary lymphoid organs, and they remember that antigen. So they're the B cell that allows your body to recognize that reinfection. So if you ever see that same virus or that same pathogen again in a month or a year or in 5 years or so on, and those B cells, those memory B cells are what allows your body to rapidly respond to that infection, and you usually clear that infection before you ever even realize you had it. And what's really cool about these B cells is that they don't require a second signal. So they don't have any kind of second signal required, which is so, and we'll talk about, I keep saying we'll talk about this later. We'll talk about what a second signal is here in a bit. All right, so again, the other major type of cell of our adaptive immune response are T cells. These also develop in the bone marrow, but then they migrate to our thymus and mature in our thymus. And in our thymus, they also undergo clonal deletion, which again, I'll talk about later. But they also undergo a process called positive selection. So T cells have to be able to recognize MHC type 1 and MHC type 2. And if they can't recognize that, they get deleted. So they're no good. So they have to kind of pass this. So we usually say that they become educated in our thymus. And what that means is that our thymus is basically checking all of our T cells that they work as they're supposed to. And if they don't work as they're supposed to, they get a signal telling them to undergo apoptosis, because we don't want T cells circulating in our system that don't work. All right, so they also have, of course, a T cell receptor on the surface, and this reacts with antigen, but it's different. It doesn't function in the same way that the B cell receptor does, because it doesn't eventually get secreted into the environment. And remember that T cells function, especially cytotoxic T cells, function mainly in cell-mediated immunity. They don't secrete any kind of antibodies, but they're really effective against cells that have some type of infection inside them, so virally infected cells, cells that have a bacterial pathogen inside, so an intracellular bacterial pathogen, cells that are damaged, cells that are metastatic or kind of pre-cancerous even. And then they're also really good, again, this isn't such a big deal for us anymore, but they're really good against multicellular what's the word I'm looking for? Parasites, right? Worms, protozoans, kind of the bigger pathogens that we don't really think about anymore because we don't usually have them. Okay. All right. So again, remember that T cells can be divided into two different functional categories. Helper T cells. We denote helper T cells with a sub H to denote helper T cells. And here's that CD denotation that I was talking about. So CD just stands for cell determinant. And so helper T cells have a very specific cell determinant on their surface, and it's number 4. So if you see a cell in your body that has CD4, this protein on the outside of it, you know that you're looking at a helper T cell. And the main function of helper T cells is to help. They really send out a very crucial second signal to other cells of our immune system, specifically our B cells and our macrophages. And they basically tell them, hey, keep going. Remember those B cells, they're as soon as they get going, they're secreting all those antibodies, and they get signals from the helper T cells to say, hey, keep going, keep going. They also stimulate macrophages to become even more phagocytic and basically ramp up their efforts to clear this infection. So helper T cells, even though it's it sounds like, oh, they just help, they kind of push people to do more things. They are absolutely crucial for our immune system to function. And we know this because CD4, very unfortunately, happens to be the receptor for the HIV virus. So human HIV, the causative agent of AIDS, can only get into a cell by attaching to this very specific receptor, CD4, believe it or not. And so what happens in a person that contracts HIV is their helper T cells basically go away. They get infected by HIV. HIV virus gets inside, replicates, destroys the cell, destroys the helper T cell, and moves on to the next cell. The next helper T cell. And so in people that have HIV, a way to mark the progression of that disease is by counting how many helper T cells they have in their body at any given time. And as that helper T cell count gets lower and lower, the likelihood that they actually develop AIDS kind of goes up. And so you guys are, all of you are too young to remember the early days. HIV is actually a very manageable disease. I don't want to underscore how important, how deadly it is, and I don't want to make light of it, but it's nothing today. It's well managed by medication now, and certainly in most developed areas of the world, it's no longer a death sentence. But in the late 80s and early 90s, or well, really through most of the 90s, it was a very, very scary disease because no one really understood what it was. But it turns out that it infects these helper T cells. Okay, so the other one are cytotoxic T cells. Remember, these are the cells that are going to be directly cytotoxic to infected or damaged cells. And they have a different marker on their cell surface, and that marker is CD number 8. And excuse me, sorry. So these cells directly cause apoptosis. They basically either send them signals that cause them to undergo apoptosis, or they just hit them with these really toxic chemicals called perforins and gramzynes that injure the cell so much that the cell dies. And these are stimulated by a cytokine signal. Remember, IL stands for interleukin. So the cytokine interleukin 2 from T helper cells. Both helper T cells and cytotoxic T cells have a subset that can become memory cells. Again, underscoring that importance of the adaptive immune response to remember upon secondary exposure to that same pathogen. Okay, our last lymphocyte, remember, not part of the adaptive immune response, but I really should just put this in the innate immune chapter. But these NK cells, NK stands for natural killer cells, are they're most similar to T cells, but they don't have a T cell receptor, and they're therefore not specific. They don't increase, they don't differentiate into other things upon exposure. They're not specific to any particular kind of antibody. What they do so what they can do is they can recognize the FC portion of an antibody. And again, I'll explain what that means here in just a bit. But they're going to recognize the FC portion of an antibody. And when they do that, they're going to launch grenades at whatever cell that antibody is attached to. So they can either recognize that FC portion, or they can recognize a cell that doesn't have MHC on the surface. Again, you're going to get sick of me saying this. I will explain what MHC is later and why MHC is important. Okay. So again, I think this was the second class in a typical class, but a good review would be to name the major types of B cells and T cells, describe their function, describe what branch of immunity what branch of adaptive immune response they play a major role in, and so on. Nice kind of summary of what we've talked about so far. Oh, wow. Two main ways. Two main ways. What would be? For me, this is an interesting. There's lots of ways, but the two to me the most important way that the adaptive and innate immune response differ is that the innate immune response is non-specific and the adaptive immune response is specific. I would say that's probably the defining characteristic of both. And then after that, I would say that maybe the other major one is that innate is fast acting and adaptive is slower to act. But we could go on, right? There's other differences. Only chordates have adaptive immune response. All multicellular organisms have innate immune response. Innate does not really differentiate between self and non-self, whereas adaptive does. It has tolerance. Lots of differences. No context at all. What are these pictures? So here I was going to ask, basically imagine at the molecular level, which of these would make a better antigen, or would you remember better, silly putty or a Lego? So keep that in mind while we talk about what the heck an antigen is, right? So again, before we put all these pieces of the puzzle together, there's yet another aspect of this that I need to kind of introduce to you, and that is antigens and what they are and what their characteristics are. So an antigen is, in a very simple way, an antigen is something that our immune system recognizes. So our B cell receptor, our antibodies, and our T cell receptor can recognize. So what does recognizability mean? Well, if we go back to this, right, silly putty is not recognized. I mean, here it's, we recognize it as silly putty, but it can take on all different shapes. It's mushy, you can flatten it out. And so it will look different every time you take a look at it. Whereas this Lego will never change its shape. It has a very characteristic shape. It has hard edges. It's big enough that we could probably, if we closed our eyes, we could probably feel it and recognize what it is again. A Lego makes a great antigen. Silly putty makes a terrible antigen. So let's talk about what these kind of things, what the characteristics of a good antigen is. So typically it is a protein or a polysaccharide, a glycoprotein, basically a polysaccharide and protein together. DNA and RNA and lipids make really poor antigens because they tend not to have a specific shape. They change their shape quite easily. Certainly, like if you think about like DNA, for example, it's held together by histones. If it doesn't have those protein chaperones, it'll just kind of twist and go out of shape quite easily. So basically, we're looking for things that maintain their shape and have consistent shape over time. And not only do they have consistent shape, but they have a definite shape. So it's very well. Again, right? So here we have like a 4x2. It has these like circular little nubbin things on the top. It has 4 corners that are hard edges, right? We can very clearly see the 3-dimensional shape of this Lego. Oh, and I think I took out a bullet. You can go ahead and This was something that I think I took out of the learning objective, so you don't have to pay attention to that. And when we talk about antigen, what we're really talking about is the epitope. And I think the next slide talks about what an epitope is better. Okay. So typically when we talk about an antigen, it's something that can be recognized usually by an antibody. And remember, antibodies are secreted by B cells. So let's imagine that this bacteria, let's just imagine that it is an E. coli, right? So E. coli, we could say E. coli is antigenic. We definitely have antibodies in our body, or B cells that produce antibodies in our body that recognize E. coli. So E. coli, you could say, is antigenic. It has antigens on it. But it's not the whole cell, right? An antibody is not going to detect DNA because DNA is on the inside. It's not going to necessarily, detect proteins that are on the inner membrane of an E. coli because, again, they're on the inside. It wants to detect, antibodies tend to detect antigens that are on the surface because those are the ones that are exposed to the external environment. And so here we can see our immune system, for example, has two different kinds of antibodies. Note the difference, like one has like these roundy ends and the other one has these sharper ends. And so this antibody is going to detect some cell surface marker on the outside cell wall, or actually E. coli is gram-negative, so the outside cell membrane of this E. coli molecule, for example. And so we can see that it's specific for this kind of roundy antigen, and then this would be the epitope. So an epitope is kind of the part that is recognized specifically by the antigen. Whereas another antibody that we produce might detect, what is this? It looks like a little epitope on the end of a pili. These little tiny little guys are pili on the outside of this bacteria. And so this antibody is only going to recognize this very specific epitope on this pili, whereas the other one isn't. This one is only recognizing this round shape that's on the cell surface itself. So even though when we talk about antigens, we can say E. coli is antigenic, or flagella is antigenic, or the pili is antigenic, what we're really saying is that there are epitopes on the flagella or epitopes on the cell surface that these antibodies can recognize. Again, highly, highly specific. You can go to the antibody store and buy you can go on a biotech company's website and there's dozens and dozens and dozens of antibodies that recognize different aspects, different antigens, different epitopes of E. coli. So they're very easy to buy. You can buy them in all different colors. They can glow in the dark. Really easy to buy. Okay. So what are these antibodies that I keep talking about? What do we know about them so far? Well, we know that they're secreted by B cells, specifically plasma cells, so B cells that have become differentiated to turn into plasma cells. And we know that they're also specific because they recognize antigenic epitopes on the outside of anything that's not self, right? So bacteria, pathogens, viruses, protozoans, all these various invaders, non-self invaders to our body. So let's talk a little bit about, we also know that they're the main effector molecule of humoral immunity. So let's talk a little bit about the structure. Antibodies are often called IG. IG stands for immunoglobulin. And this is because when scientists were trying to figure out what these were, they would basically take serum from an animal. They would isolate the proteins and then run those proteins on a gel to separate them by size. And the portion that came out in the gamma portion of this protein array was the proteins that seemed to have some type of immunological function. So they called them, so it was the gamma fraction of these immunoglobulins, and so we or of these proteins, and we knew that they had an immune function, so we called them immunoglobulins, or Ig for short. So these proteins and they're proteins, have two portions. They have, or two areas, I should say. The FAB portion, which binds to the antigen, that's a portion that is responsible for binding to antigen, and the FC portion, which basically carries that signal to other cells of the immune system. So let's talk about the structure a little. And I kind of think of antibodies at like a tool, kind of like an interchangeable tool. On one end, like if you think about a drill, the drill bit end, goodness, how many different adapters are there for a drill bit, right? Hundreds, right? Well, it turns out that antibodies have billions of different combinations. But you think about a drill bit, like it has all of these different combinations that you could put on one end to make it recognize different, different screws or different nuts or bolts or whatever they are called. But the other end, the FC end, always stays the same, right? And basically serves the same function. It's where you hold it and it's where you charge it and so on. And so that's kind of what antibodies are like. They have one end that is highly, highly variable. And this is the spot that we want to be variable. We want it to bind to as many different kind of antigens as possible. And so this end is called the antigen binding site. I'm getting ahead of myself. Hold on. So let's first discuss the regions of the antibody. Remember, we have two regions, the FAB region and the FC region. FAB region contains the variable region. So we can see the variable region are the blue and green regions. The constant region are the red regions and this like light pink region. And then it's also it is also made of 4 different chains. So 4 different kind of proteins come together to form this antibody. And you can see the 2 heavy chains are kind of in the middle. And then we have 2 light chains that are on the outside. Notice that they're held together. There's like these tiny little bridges. They're held together by disulfide bonds between what amino acid is that? Methionine, I think, or no, cysteine. Cysteine or methionine has sulfur in it, and they can create these disulfide bridges that really anchor the proteins together and make sure that they behave as a unit. So the heavy chains have 4 different kind of flavors: gamma, mu, alpha, delta, and epsilon. It's easier just to call them by G, M, A, D, and E. And then the light chains have 2 variations, kappa and lambda or K and L. So why all of this, right? What's the point of this? Well, look, if you notice, the FC portion is only made up of the heavy chain. And this portion is not going to change based on the class of heavy chain. Notice there's 5 different classes of heavy chain. So that's really going to determine what the FC portion looks like. Whereas the top, this variable region, is made up of both the heavy chain and the light chain. And really what we're really looking at in this variable region is the very end of it. Because this is the end that is going to interact with epitope. Right, so this determines that it can bind to some specific epitope or not. And this is the site that is highly, highly variable. And we want that because we want as many different antibodies available to our body as we can. So let's talk a little bit more about these different classes. So based on the heavy chain, remember there's 5 different heavy chains, G, M, A, D, and E. So let's talk about these different categories. So this is going to determine the kind of antibody that we have. So there's 5 major classes based on the structure of that heavy chain. So the first one is Ig gamma or IgG. And this is by far, like, this is the one you want. This is the one that is best at recognizing epitopes. It's really long lasting. It sticks around for up to a month. This is also the one that can cross the placenta. So this is the one that basically protects a developing fetus. And then not only does it protect the fetus while it's in the womb, but after birth it can stick around. I mean, I think the half-life on an IgG is like two or maybe even 3 months for IgG. So I guess that's possible if those coronavirus antibody tests are for IgG. It could maybe potentially go back even further. But more importantly, so going back to the function of IgG, it after birth, it's going to protect the baby for at least a month or two after birth as well, which doesn't sound like a big deal, but it's a super big deal because babies don't have adaptive immunity. Remember, babies only have innate immunity when they're born. So the only adaptive immunity they have is the antibodies that are given to it by the mother. So let's see. What else is important about IgG? So remember that complement is the branch of the innate immune system. There are those tiny little proteins that kind of circulate throughout our blood and get turned on, and they create this complement cascade. And one pathway of activation of complement is the antibody pathway, and that is activated by IgG. So IgG is, I want to say IgM does this also, but IgG is one of the antibodies that will activate, is really good at activating complement. It also can act as a big red flag to phagocytes. So basically what happens is it binds with its variable region to the bacteria, and then that Fc region sticks out into to the external environment and acts like a big red flag to recruit macrophages to come and eat that bacteria. I have to say, it really stinks here. So I'm in my apartment, and I think the red tide that's making all those really cool bioluminescent pictures that are all over Instagram right now, it's starting to die, and it really, really stinks here, and it's annoying. I'm sorry, I had to get that off my chest. Okay. So the third big thing it does is it's also the antibody that recruits natural killer cells. So if IgG is stuck to one of our own cells, or any cell really, it's going to act as a recruiter and a big red flag to recruit natural killer cells. And once natural killer cells bind to that Fc portion of IgG, it's going to kill whatever that IgG is attached to. Let's see, we haven't talked about valence. So valence basically means how many binding sites it has. And notice that, so if it kind of has this basic Y shape, it can attach an antibody there and there. So when we talk about a valence, it means that it has a valence of 2. Now contrast that with IgM. What's the valence of IgM? 1, 2, 10. Right? Because what happens here is it basically forms a pentamer. And that pentamer, so that's 5, and each one has those 2 binding sites. So we would consider the valence of an IgM antibody to be 10. IgM is not great, but it's early. It's the first antibody that is produced and secreted upon initial antigen exposure. And it's really good, as you can imagine, with all 10 of these binding sites. It's really good at like trapping and immobilizing and agglutinating. So agglutination basically means like clumping together of bacteria or of toxins, and then that creates that agglutination, and immobilization will start to recruit phagocytes, basically macrophages, to come to the site. That's what I thought. It was also it's also pretty good at complement fixation. It activates what is that, C3, the C3 convertase, to start that complement cascade. Okay. I think yeah. So this is basically what I've been going over. This is a really nice table in your book. It's table 15.1, that summarizes all the various antibodies and their main function. And I'll talk about IgA and IgA, IgD, and IgE here in the next slide. So IgA, the cool function of IgA is that, well, it's a dimer, so it has a valence of 4, but it's really the main mucosal secreted antibody. So any of the mucosal secretions that we have, if there's antibody in it, it's IgA, and it's also found in breast milk. So if for example, there was a question on the test that asked you if what kind of immunity does a baby have? Of course, the obvious answer is innate immunity, but it also has IgG left over from the placental crossing over, and it could also have IgA from that breast milk, and that serves a critical kind of protective function in the early months of an infant's life before it can start developing its own natural adaptive immunity. IgD is, let me think about IgD. This one is so this is the one I don't really know about. This is the one that is, it's really not well understood. It kind of has, let me go back to the table. I'm pretty sure this is the one that it's probably like an evolutionary holdover, and its function isn't really well, oh yeah, see, look at me. Functions in blood not well understood. There we go. So I don't think that IgD is very much studied. Again, very, very small. If you notice here, it's less than 1% of the total immunoglobulin fraction in our serum. So it's a poorly understood antibody, but IgE on the other hand is unfortunately very well understood because IgE is the major player in allergic reactions. So basically what IgE does is it binds to mast cells, and we didn't really talk much about mast cells. They're part of our innate immune response. And they also bind to basophils, and essentially what happens is when these mast cells and basophils get attached to IgE, they just start pumping out histamines. And so IgE, whenever someone has an allergic response, the antibody that you find in their system is IgE. Okay, so let's talk about what these antibodies actually do. I touched on this briefly, but I want to kind of formally go over this. In fact, I think I'm going to take a break here and divide this lecture into 2 parts so that it's not a super long video. So I'm going to take a pause here, and then I'll start right up again here in just a second.